So, just to reiterate and play a little animation, is that here's the one cycle of a typical polymerase chain reaction. You first denature DNA, and your double helix comes apart. Then you anneal primers. You will have a forward and reverse primer. In the first step, the forward primer will anneal to the antisense strand of your template, and the reverse primer will anneal to the sense strand. So in order for it to be able to elongate. So your primers are always going to be in this orientation. One will be, the forward primer will be on the sense strand that you care about. It will be basically the first 18 or 25 base pairs of whatever gene you're interested in, will be your primer, or a little bit more, 5'. And then the reverse will be on the antisense strand. It will be complementary to the antisense strand. A lot of people make the mistake to put both their forward and reverse primer on the sense strand, but then you're getting totally wrong reaction. You will not get the proper reaction that you're looking for. And then, so once the primers have annealed, you will elongate the primer and create more DNA using DNA polymerase at 72. And then you will repeat this 1, 2, 3 PCR cycle about 25 to 35 times to get this massive amplification of the DNA that is nestled between these two primers. The rest of the DNA of the template will not be amplified, just the part here. So in the first cycle, you will see that the DNA polymerase will continue going. But as you've seen in the previous slide, the more PCR cycles are performed, the more defined the product becomes until overwhelming majority of the DNA in the test tube where the PCR reaction has been performed is all the same length. So this is again all the steps of the DNA of the PCR reaction, and you can technically do this whole process with 3 water baths, constantly moving your tube back and forth. And when the polymerase chain reaction was originally performed, people did set up 3 water baths and either a robotic arm or a graduate student to move the tubes from one bath to another, until several companies, among them Bio-Rad, had optimized these these thermocyclers that can be just programmed to go through the cycles 35 times. And they're not cheap, but they're not all that expensive nowadays. They're about $4,000. So what I wanted to show here is that PCR amplification occurs with 2 primers annealing to the opposite strands. So you guys can see in this case, the forward primer is binding to the antisense strand, and the reverse primer is binding to the sense strand. And then after the 3rd cycle, the majority of the product in the test tube is DNA fragments that are flanked by the 2 primers, as opposed to the remaining DNA molecules that have been amplified from the template and are much longer. All right. So another thing I wanted to talk about is how the field of DNA polymerases that are used for PCR has evolved since Kerry Mullis and others chose to use Taq polymerase for the PCR reaction. The problem with the original Taq polymerase is that even though it's very stable at high temperatures, it does not have 3' to 5' exonuclease activity, and so has a very high error rate. So you guys can see here the Thermos Aquatica's Taq polymerase lacks the 3' to 5' exonuclease activity. You guys can see that the little red square that is marking the exonuclease site is gone. So people since then were working hard on identifying other DNA polymerases from thermophilic organisms to see if they would be any better than Taq and have a better proofreading ability, or at least any. Several ones have been identified. So there's Thermococcus codecransis, and the enzyme I'm more familiar with is PFU polymerase. It's isolated from Pyrococcus furiosus, which is a bacteria, which is not even a archaea, it's an archaea isolated from deep sea vents around Italy and volcanic mud in Italy. And PFU does have proofreading. It has a 3' to 5' exonuclease activity. But because it has an exonuclease activity, it can degrade primers some, so usually you add a little bit more primer and start the reaction at high temperature. First, it's a hot start polymerase. And it's also not as fast as the Taq polymerase. So you guys can look at the relative processivity values of Thermococcus aquaticus and Pyrococcus furiosus. You can see that Thermus aquaticus polymerase is 6 times faster than PFU. There's also other issues that I will not discuss today. So even though Taq makes mistakes, and a lot of them, it's still a really great polymerase that you use for PCR applications where you just need to detect a certain stretch of DNA, but you don't need to then take this DNA and, for example, use it in cloning or for protein expression and so forth. You just need to show that that particular DNA fragment was present in the DNA you were probing. But because Taq was making lots of mistakes and PFU wasn't very fast at the PCR reaction, people have been trying to modify the PFU protein to see if they could do any better. So one idea was to improve the processivity of the PFU polymerase in order to make it stay on the DNA longer without the clamp, and so be much better at elongating. Because one of the problems is that polymerase PFU tends to fall off its the single-stranded DNA and it takes a while for it to get back on, so it doesn't make quite so many copies per second as Taq does. So people in New England Biolabs company fused PFU with a DNA binding protein that was also thermostable and fully functional at 95 degrees. So it got this little Superman cape that allowed it to hold onto the DNA. Remember that the DNA molecule goes through right about here. And the reason I drew it as a little cape is because when you actually look at the predicted crystal structure of the fusion PFU, so this is the PFU part and this is the DNA binding part, you can see that it does sort of look like a little Superman cape on the little polymerase. And adding this DNA binding protein that allowed the PFU to stay longer on a template dramatically increases processivity. Also, because the enzyme used for this fusion was not the original Pyrococcus furiosus DNA polymerase, but a mutated DNA PFU that had even better proofreading activity. The people from New England Biolabs really created a super DNA polymerase. So the processivity of the DNA polymerase, the fusion DNA polymerase, is now better than the Thermus aquaticus Taq polymerase. So 6 versus 10. It's not that much better, but it's much better than what the original PFU was able to crank out, which was barely 1. So hopefully this slide shows you guys how the DNA technologies field is constantly being optimized and improved, and what kind of things you can do to make something like a DNA polymerase work for you even better. So fusion polymerase is not something found in nature. It was specifically created for improving the processivity and the error rate of polymerases that are used in vitro in PCR reactions.
There was a time when to amplify DNA, you had to grow tons and tons of tiny cells. when along came a guy named Dr. Carey Morley's.
Said you can amplify in vitro just as well.
Just mix your tablet with a buffer and some primer. Nucleotides and polymerases too.
Denaturing, annealing, and extending.
Well, it's amazing what heating and cooling and heating will do.
PCR, when you need to detect mutation. Detect mutation. PCR, when you need to recombine. Recombine. PCR, when you need to find out who the candy is. PCR, when you need to solve a crime. Solve a crime. PCR, when you need to detect mutation. Detect mutation. PCR, when you need to recombine. Recombine. PCR, when you need to find out who the daddy is. PCR, when you need to solve a crime. Solve a crime. PCR.
To all the scientists out there doing PCR, Biorad salutes you with the all-new 1000 series thermal cycling platform.
Hi. So we're going to continue the DNA Technologies lecture, and right now I would like to talk about various things that you can do with the PCR, with the PCR-based technology. So you can see here this laundry list right here includes sequencing, identifying individuals, identifying related individuals, creating novel DNA sequences for synthesis, for cloning and synthesis, or just amplifying a chunk of DNA from the large genome, like we try to do right now with the fluorescent protein from a coral that I used as an example. So I want to talk to you about this lecture question here. Here, because you guys hopefully remember that typical PCR reaction requires 2 primers. If you guys look at this question and pause this lecture and think about it yourself, what is the primer combination that will copy the entire coding region of this green fluorescent gene? So I'm going to take a... Okay. Hopefully you have paused it, so let's go over the answers. So the answer is that you need to amplify the entire coding sequence of this fluorescent gene from a coral. You would need to have this primer B and this primer D. Or if you had a reverse one of C, that would work pretty well too. But between B is beginning here and D is beginning here, and so the region that you will amplify will be this. B and D, so if you use the sequence A and C, you can see that A is directed this way and C is directed this way, so the polymerases will be moving into 2 separate directions, and it will not be possible to have an exponential amplification of the product. And if you would take C and D, these primers are oriented towards each other, so there will be an exponential amplification, a PCR chain reaction going on. However, it will not be of the whole gene. So because C and D do not span the whole gene, it just spans the C terminal of the gene. Now, as I just mentioned, PCR can be used to distinguish one individual from another. And that's because of certain genomic irregularities we all have inside our DNA. Our DNA contains several areas of short tandem repeats, and the length of these short tandem repeats will differ from one individual and the other, and will be very similar in related individuals. As a result, you can amplify the areas flanking. You can have primers that flank these short tandem repeat areas that we all have in our genomes, and then amplify the areas using a PCR, then run it out on a gel electrophoresis and see what is the length of this area. Slightly longer STRs will run higher, and slightly shorter will run lower. And by comparing the STR areas of a paternal, maternal, and a child's DNA, you will be able to make conclusion whether the parents match the child. So, and since you can extract DNA from just about anything organic, you can get enough DNA for such a reaction from hair, blood samples, saliva samples, etc. Because a PCR reaction will amplify the amount of DNA you start with about 1 million-fold, so you can start with very little DNA to begin with. And as I said, once the PCR reaction is finished, you amplify the product, you separate the products on gel electrophoresis, and you will see how many bands and what size, how the bands match. So let's go over an example like here. Let's say you have a forensic sample from a crime scene, and then you have 3 different individuals that you took DNA samples from via saliva or whatever. And in this example, 4 different PCR reactions have been run. Actually, kind of depends how you want to do it. So you can either run 3 separate PCR reactions for each of these colors, or because the primers for red are the same and the primers for green are the same and the primers are for blue, but because the STRs for the 2 chromosomes of each of the suspects will be different, 2 different size products will always be produced, one from your maternal chromosome and one from your paternal chromosome. Now, if you usually such reactions are run either in separate individual tubes, or you can have a multiplex PCR reaction where the annealing temperatures of all of these 6 different primers have been optimized to bind at the same annealing temperature, and the protocol has been troubleshot. So usually when you actually do forensic PCR or paternity PCRs, you will run multiplex reactions in one of your test tubes, and the PCR primers have been fully optimized for that reaction. Anyway, that's technical details. So you can see that there's a different fluorescent primer that will allow you to distinguish between which of these products came from the red STR, green STR, or blue STR. And then you run it all in a gel. It's going to be either electrophoresis, or you might be using gel capillaries, depending on the size. And you then look at this gel and try to figure out so if you're trying to analyze which of these 3 individuals matches the forensic sample, you would be looking for perfect match. So in this case, you guys can see that B, the PCR reactions in the B lane, perfectly match the PCR reactions in the F lane. So just think that the DNA sample coming from the F and from the B is identical. If you were instead looking for paternity, you would not look for identity, because father and son will not have identical STR profile, but will be slightly different. Sorry, this is a misspelling. It's not SNP, it's STR. But you would look for matching bands on the 2 gels. In this case, if you want to see who matches the paternity of F, we already know that B is identical to F. So let's pretend F is forensic and we'll just discount that. So if we're trying to find B's parent, is it A or is it C? You can see that in C, hardly any of the bands match that of B. However, in A, you can see that the red bands both match, and one of the bands in C, and one of the blue bands match between A and B, and one of the green bands matches in A and B. So at least half, suggests that half of the chromosomes match. So this individual B has this chromosome with this particular SNP, and so does individual A, but their other chromosome, because he inherited, the individual B inherited from his mother, doesn't need to be matching the individual A. So you guys can see that these two individuals share many SNPs, in fact half of them, and it is by just sheer luck that both of the copies of 10 and 30 are the same. So these two individuals are related, while B and C are not related. And this both STR genotyping using PCR and other approaches have been used to identify individuals. One of these individuals was Osama bin Laden, who was, and this was the approach that the CIA used. So they knew that bin Laden's sister was treated and died from brain cancer in Boston, and were able to obtain a DNA sample from the brain tissues that the hospital still had on file. And CIA operatives had tracked down bin Laden to a particular town in Pakistan, but they needed confirmation before they could launch an operation against that particular household. They needed to be very sure that the person inside was Osama bin Laden. Now, because because vaccination programs have been performed in Pakistan to stave off the reappearance of polio in the region, especially in the Abbottabad region, the CIA had recruited one of the Pakistani doctors who was part of the vaccination program to inject somebody to have a hepatitis vaccination program in the neighborhood, and they saved all the syringes from the vaccination program, and then the blood from these syringes was extracted, and DNA the analysis of the DNA was performed using shorthand and repeat approach, SRP. And they were able to match bin Laden's blood to show that there was a person in the compound that was matching the DNA brain sample that they had from bin Laden's sister. As a result, the attack on the Abbottabad compound was launched. Osama bin Laden was killed, but the news about this leaked, and the news about the approach leaked, and as a result, the vaccination campaigns and a lot of vaccination of polio vaccinations in Pakistan were stopped, and a lot of vaccinating doctors and nurses were killed by the insurgents in retaliation.